Hey there, science fans! Have you ever wondered how scientists study things too small to see? We're talking about the microscopic world of DNA and proteins. It's pretty amazing. Special lab techniques allow us to zoom in and unlock the secrets hidden within these tiny structures. These techniques are like superpowers for scientists. We can amplify DNA, detect specific proteins, and diagnose diseases. In this exploration, we'll delve into two essential techniques: polymerase chain reaction, or PCR, and enzyme-linked immunosorbent assay, also known as ELISA. Get ready for an exciting journey into the fascinating world of molecular biology. It's time to unleash your inner scientist. Let's start with PCR, a revolutionary technique that's like a DNA photocopier. Imagine wanting to study a specific gene, but you only have a tiny amount of DNA. PCR comes to the rescue by making millions of copies of that specific DNA segment. Here's how it works. First, we mix the DNA sample with special ingredients. Primers act like bookmarks, attaching to the start and end of the target DNA segment. Then, an enzyme called DNA polymerase acts like a tiny construction worker, building new DNA strands using the original as a template. This process happens in a machine called a thermal cycler. It heats and cools the mixture in a cycle. Heating separates the DNA strands, and cooling allows the primers and polymerase to bind and build new strands. Each cycle doubles the amount of target DNA. After several cycles, we have millions of copies. PCR is like magic, but it's real science. It's used in so many fields, from diagnosing diseases to solving crimes and even tracing ancestry. Pretty cool, huh? Now let's dive into ELISA, another essential lab technique. This one is all about antibodies, those amazing proteins in our immune system that fight off invaders like viruses and bacteria. ELISA helps us detect and measure these antibodies, or the specific substances they bind to, called antigens. Imagine trying to find a single grain of sand on a beach. That's what finding a specific antibody or antigen can be like. ELISA makes it possible. We start by coating a surface with antibodies specific to the target we're looking for. Then, we add the sample, which may contain the antigen. If the antigen is present, it will bind to the antibodies. We wash away anything that doesn't bind. Next, we add another antibody that is linked to an enzyme. This enzyme will cause a color change only if the antigen is present. The amount of color change tells us how much of the target substance is present. Pretty neat, right? ELISA is like a detective, sniffing out specific molecules in a complex mixture. This technique is used to diagnose diseases like HIV and COVID-19, test for food allergies, and even detect pregnancy hormones. Section 4. Mastering the technique's the key to reliable results. We've learned that PCR amplifies DNA and ELISA detects specific molecules. But why are these techniques so important? Well, they're the foundation for many scientific discoveries and medical diagnoses. Mastering these techniques is crucial for obtaining accurate and reliable results. Imagine baking a cake. If you don't follow the recipe carefully, you might end up with a disaster. The same goes for lab techniques. Precise measurements, sterile conditions, and careful execution are essential to avoid errors and ensure accurate results. Proper training and practice are key to mastering these techniques. Like any skill, the more you practice, the better you become. It's like riding a bike. At first, it seems tricky, but with time and practice, it becomes second nature. Section 5. Practice makes perfect, honing your lab skills. Mastering lab techniques like PCR and ELISA is an ongoing journey. It requires patience, attention to detail, and a passion for scientific exploration. Remember, even experienced scientists were once beginners. Don't be afraid to ask questions, seek guidance from mentors, and embrace the learning process. The more you practice, the more confident and proficient you'll become. And who knows? Maybe one day you'll make a groundbreaking discovery using these very techniques. So, roll up your sleeves, put on your lab coat, and dive into the fascinating world of molecular biology. With dedication and practice, you can master these essential lab techniques and unlock the secrets of the microscopic world. Section 6 up next, exploring the world of lab instruments. Congratulations on learning about PCR and ELISA. You're well on your way to becoming a lab whiz. But our journey into the world of science doesn't stop there. Next time, we'll explore the amazing instruments that scientists use in the lab. Get ready to discover the tools of the trade, from microscopes that reveal hidden worlds to centrifuges that spin at incredible speeds. Stay curious, keep exploring, and never stop learning.